This is my great pleasure to welcome Dr. Julie PANNEQUIN. Julie is a research director at CNRS, and she directs the team called Signaling Plasticity and Cancer, which is located at the Functional Genomic Institute in Montpellier in France. Julie has recently developed a project on cancer stem cells and their impact on resistance to chemotherapy. Julie is also interested in the translational mechanisms leading to the plasticity of these cells. And at last but not least, her team also investigates the role of circulating tumor cells in early dissemination and dormancy. And we are really happy to hear you, Julie, and speaking about early dissemination in colon cancer. Thank you very much.
Ça marche pas comme ça avec ça. Si ça marche. Okay. Okay. Hello everyone. Thank you very much, Fatima, for the kind invitation. I'm really impressed to be here and talking about after all these great people. I'm gonna try to be not as good, but I'm going to try to be at a good level. Anyway, this is my team, beautiful team in Montpellier. And I want especially highlight the fact that here there is Zainab that is in the audience and that has done most of the work that I'm going to present today. So as Fatima said, we are doing translational research. We are working on colorectal cancer, and the main issues in colorectal cancer are recurrence and metastatic disease. So we are interested in chemoresistance, plasticity, and dissemination, and today the focus of my talk will be dissemination. Why? Because, as you have heard already, metastatic disease is an incurable disease. And in colorectal cancer, if you have 100 patients diagnosed with colorectal cancer, there will be only 3 surviving after 5 years following the treatment. So there's definitely something to do. And the project that I'm going to present to you today starts with this very simple question: when the cells start to disseminate. Is it something that follows the classical model that we have learned at school, that you have the first mutation, an adenoma forming, and then accumulation of mutation to form a carcinoma that will become invasive and go into distant organs through the circulation? But in parallel, maybe you have heard that there is also an early dissemination that occurs at the very early stages of tumorigenesis. So this has been really well shown in melanoma, in breast and pancreatic cancer, recently in gastric cancer also, but in colorectal cancer there was not much. So we have decided to tackle this issue by generating a mouse model that will mimic the very first step of the intestinal tumorigenesis. And just to remind you, the first hit in this tumorigenesis is, in the vast majority, is a mutation on APC. So we have generated a mouse model that specifically in the intestine will have a fluorescent, will express a fluorescent protein, RFP, and will have a mutation on APC. So upon tamoxifen, the whole intestine will be red, and there will be the development of some intestinal adenoma, as you can appreciate on this photo. Okay, just importantly, the control mice of this study are the mice with a red intestine and treated similarly with tamoxifen. And all the mice have been sacrificed between 3 to 4 months at the stage where we are at the early adenoma, validated by pathology. So we never reached the stage of adenocarcinoma. So the first thing that we have done is to see whether we could see in the adenoma, in the vascularization of adenoma, we could see some cells that are already circulating. To do that, we have performed some very basic intravital live imaging. And I hope, yes. Yes, I missed it already, but I hope you have seen. So this is an adenoma. We are on an adenoma. We see the vascularization. The light is the fluorescence because it's in the intestine. And as you can see, there is a cluster of cells that goes in the circulation, and that is fluorescent, though suggesting that they come from the intestine. Will this cell be able to reach distant organ to create metastases? I have no clue, but at least they are circulating. So the next obvious question that we ask is whether we could detect some cells at these early stages in distant organ. And for this study, we have decided to focus on the liver, which is the main organ for metastases in colorectal cancer. So the answer is yes. By immunolabeling, we could detect specific staining of RFP, meaning that these cells are intestinal. And we have also done some tissue clearing in the collaboration with Ali Ertug in Germany. And we could see that we have some intestinal cell, very rare, either in small cluster or isolated. And we were able to quantify the number of cells. And in the whole liver, we never reached 500 cells. So it's really rare events, but still it's an event that occurs. I didn't tell you, I forgot to tell you that the mice that we generated never form metastases, never develop metastases. Even if you keep them for more than 1 year, you never see metastases. And if we inject the adenoma into nude mice, we never have the development of tumor. So these cells are not tumorigenic. So the question is why these cells are going so early in the liver if they are not making any metastases. So to answer this question, we use the global approach of single-cell RNA-seq in the liver of wild-type mice and in the liver of mice that have intestinal adenoma, but for me, I was expecting that they have a complete normal liver. And as you can guess, as you can see, not guess, the landscape is completely different. And if you analyze this single cell, you see that the most striking change is an enrichment in myeloid cells, and including macrophages and neutrophils. So we then validated this at the protein level by using site of Hyperion. And once again, we have a huge enrichment of macrophages and neutrophils in the liver of these mice that have intestinal adenomas. So we have validated this data in several complementary models using bigger cohort of mice, using different markers, and each time, as you can appreciate here, macrophages as well as neutrophils are highly enriched in the liver of this animal at very early stages of intestinal tumorigenesis. As you can see here on the staining, this staining is completely diffuse and is not restricted to the presence of early disseminated cells. So we next wonder whether at the local state, around the disseminated cells, if there were a transcriptional program that was different. So to answer this question, we have performed some spatial RNA-seq, and we have picked some area that do contain early disseminated cells in the liver, and some area that do not contain these rare cells. And then we analyze this, and what we found is that indeed we have a different program when we are near the RFP-positive cells compared to when we are far from them. And when we analyze them, one of the highest activated program was the TNF-alpha signaling via NF-κB. And you may know that NF-kappa-B is regulating lots of things and is regulated by lots of different processes. But what was interesting here is the cytokines. So we next wonder, are there some cytokines or circulating factors that are differently represented in the plasma of wild-type mice versus mice that have these intestinal adenomas? So, of course, the answer was yes. And we did a cytokine array and then quantified the top candidates by ELISA. And we found these circulating factors that are highly associated with neutrophils and macrophages in the literature. So that was kind of good to know. But the next step was to say, okay, so it seems that the adenoma secretes these circulating factors, and this will recruit the myeloid cells in the liver, and this is a systemic effect. But the next question was, but these early disseminated cells, can they do the job alone? So to answer this question, what we have done is to inject adenoma cells, a very few number of them, 1,000 of them, into the liver of wild-type animals. Here, there's no polyp at all, and in comparison with PBS. And after 15 days, we look at the macrophages and the neutrophils. And here we see that we have a partial enrichment in these two subpopulations, strongly suggesting that these cells alone can partially recruit the myeloid cells, but it might be in synergy with the circulating factors that we have identified. So the next question was, okay, what's the point? You have these cells, you have these mice that develop polyps. This polyp will disseminate, some cells will disseminate, go into the liver, remodel the liver, but for what? So because, as I told you, these mice never develop metastases, we decided to mimic the latest stage of the intestinal tumorigenesis, which is the development of metastases. So we inject carcinoma cells into the spleen to let the cells go into the liver of wild type, normal liver, or liver that has been previously remodeled. And we wanted to compare the ability of the cells to colonize this organ. So first cohort of mice, we sacrificed them 15 days after the injection. And here it was black and white. It's only when the liver has been remodeled that we can see some micro-metastases. If we leave another cohort of mice a bit longer, this time 30 days, we can see that in the control we have we have metastases as expected, but it's nothing compared to what we have when the liver has been previously remodeled. So the question of the reviewer was, okay, that's quite nice, but show us that this remodeling is the culprit, is responsible for the facilitation of the colonization. So to answer this question, we decided to deplete the neutrophil. So we did exactly the same thing, but 1 month before the injection of the carcinoma cells, we deplete the neutrophil with anti-LI6J, and we looked whether we had an effect on the colonization. And here we have first validated that the depletion worked with the spleen, as you can see. And here the result was striking. We have no more metastases in the liver when we have depleted the neutrophil before during the remodeling. So we arrived to the point that we propose, at least in mice, that there is an early dissemination in the intestinal tumorigenesis, which consists of some adenoma cells together with cytokines that go early into the liver to recruit myeloid cells. And later on, there will be another wave of dissemination with cells that have more mutation and that are more aggressive and capable of forming metastases. And this pre-metastatic niche preparation is facilitating the colonization and then the metastasis development. So, of course, before finishing, we wanted to know whether this was relevant for clinic. And we collaborated with a local hospital. And because we could not have access to the liver of patients with intestinal adenoma, we took the blood of patients that underwent colonoscopy. And first, on this blood, we confirmed that we have another representation of the same circulating factors than the one that we have identified in the mice. And what we have done is that we have succeeded in isolating and make some cells surviving in culture. So cells that come from the blood of patients with intestinal adenoma. And we have shown that these cells were epithelial and intestinal because they express CK20. And for 3 of these patients, we have been able to amplify enough to have RNA-seq. And what we could see is that these 3 patient sample express various markers of epithelial and intestinal cells. So with that, I'd like to thank all these beautiful people, and especially, again, Zainab for the amazing work that she has done in the lab, Tina, Caroline, and Laia, and Guillaume, who has initiated this project, and all these people, all the funding, of course. And before finishing, I'd like to let you know that we have built a network on tumor heterogeneity and plasticity in France. And we are celebrating our 10th anniversary. So you are all welcome to register. There's a really nice list of keynote speakers. And the early birds are until the 24th of February, I think. So be quick to pay less. Thank you very much.
Very interesting. Do you have any idea of how long the adenoma cells stay in the liver?
No. We have sacrificed mice more than 1 year, as I said, more than 1 year old, and they were still here. Wow. And there were still a couple of them not proliferate. I mean, I don't know if they I don't think they proliferate. It's only cluster of small cells. They stay. I don't know. What happened in patients is that the polyps, often they are removed. So the very important question is whether this is reversible, if these cells just disappear after when there's no secretion of factors. But it's really hard to answer this in the mice because the APC-mutated mice always form new adenomas, and then you cannot do that.
Thank you, Julie. Very, very nice. Do you think that this is a liver-specific trait, or is it common? So this dependence on neutrophils is...
Yeah, So that was a question of the reviewer also. So we have done this in lungs because lungs is the second organ where you have metastasis in colorectal cancer, and there is this tendency, at least in the enrichment in myeloid cells. But we have also looked at kidney and pancreas, where normally there's no metastasis and there's no enrichment. So maybe, I don't know, it's maybe a bit ambitious to say that, but maybe this is a process that participates to the seed and soil, because it's not something that is completely systemic and there's an inflammation everywhere. It seems to be quite specific.
Yes. First of all, congratulations to be, let's say, courageous enough to do this kind of research and to actually go against some aspect of the dogma. Okay, so I think congratulations. And I have a specific question. The polyps that you get, they are both in the small and in the large intestine.
Yeah, but we have done exactly the same thing in AOMDSS model that are only in the colon.
So you pick up only those from the colon.
What? Sorry, what
no,
was
in the mice,
I stopped
to
you.
do the experiment in the mice.
Yeah, yeah.
So you pick up the polyps or the adenomas.
Yeah.
In the colon part of the gut, or in the small intestine part of the gut?
To do what? To do what? Because for the most of them,
I forgot.
Yeah.
To do you inject cells.
Ah, okay. So when we inject to see whether these cells were able to.
Yes. Because they don't have the same physiology.
Large intestine. Yeah.
It's from the large intestine.
Okay.
Not from the colon.
Not from the small, you mean?
Not from the small.
Okay.
And not from the...
Patients, no.
No, but normally in the mice.
Yeah, they develop a lot.
They develop mainly in the small intestine, believe me.
Yeah. But that's why I said that, we have done exactly the same thing with AOMDSS that develop mainly in the colon, and we have the same kind of results. So.
And then you said this adenoma never give rise to metastasis. Okay. But do they progress to the next step? For instance, RAS mutation and others.
So in our hand, we never had a carcinoma because the polyp become too big and you need to sacrifice the mice.
Okay.
But I think depending on the health status of the animal facility, sometimes people can get carcinomas, but to my knowledge, they never develop metastasis.
No, that's I believe.
Yeah.
It's very hard to get metastasis.
But I think some of people they describe that they can go toward. But for us, we really wanted to stop at the really early stages to make sure that we are not at the invasive stage.
And do you think your observation may apply to other benign tumors from other organs?
But it has been shown in very early stages of breast, pancreas, and melanoma. And very recently, there's this study on gastric cancer that showed that even the normal gastric cell circulates. To do what? I have no idea, but yeah.
As well as exosomes.
As well as exosomes, yeah, I guess.
Okay.
I just wanted to make a comment that you could see pre-malignant stages are already influencing the distant future metastatic organ even before they're a cancer cell. So if you have a pro-inflammatory situation like IPMN or PANNEQUIN, it's already republished that. It's already attracting neutrophils in the liver and completely changing the metabolic profile And in agreement with what you see, if we use osteosarcoma, the neutrophils are recruited in the lung. They're there before you get metastases. And if you use a liver metastatic model, they're only in the liver, they're not in the lung. But colon has both, like you say. And so you see neutrophils in both organs. So like you can condition an organ and then use a metastatic tumor cell, like you say, and make it go there. So if you do preconditioning, like we showed in our paper, you can use a lung tropic exosome, precondition, get the neutrophils in, and then add a bone tropic tumor that never grows in the lung, and it will grow in the lung once you condition the neutrophil part. Neutrophils are absolutely critical. I'm not reviewing your paper, but we find exactly the same thing that you're reporting.
Okay.
Okay. Beautiful work, Julie. I have two questions actually about the CTCs. You said that the CTCs from patients, they do express MEX3A. So I was wondering whether they can be considered as cancer stem cells. And the second question is, do you have any idea of the homing process that is involved in the recruitment to the liver or lung for the cells?
So alors, first question. This is RNA-seq, this is bulk. We have, I mean, it's not a perfect RNA-seq. It was really hard, really challenging. And we think, I mean, we are sure that there's a mix of immune cells with circulating cells that survive together. And we have RNA-seq, all of them. The next step will be to do some single cell, but we were not ready. Do they have some stem cell property? Might. I have no idea. I don't know. It's possible. Voilà. The second question, I can't remember. What is it?
The second question is about the homing process for the CTCs to go to the liver or the lung. Do you have any idea of the mechanisms involved?
No, we have now deciphered the mechanisms how the early disseminated cells recruit the myeloid cells. It's through TIMP1, one of the most enriched circulating factors. But how these cells go, we have no idea. We just focused on the liver, and yeah, it's really interesting to know what's happened before, but yeah, we don't know.
I was wondering if you compare the cells from the primary site, the first disseminating cells at the primary site inside the circulating blood, and after the ones that succeed to be in the liver. Okay, so when we started this project, it was exactly the ambitious idea that we had. It's easy, the intestine is red, so we're going to be able to follow and to sort all the red cells in all the animals. But at the end of the day, you stay 3 hours in front of the cytometer and you have 20 cells and you don't know really what to do with them. You know, we have bought a lot of kit to RNA extract, but it never worked really. So yeah, ideally it would be so good to have that. What is good from the patient is that we have the section of the polyp of the same patient. So we're going to be able to extract the RNA from the polyp and to compare to this. But to the early disseminated cells, could not. And if you do some like tracing to, when you inject your 100 cells, you can add a tag for the 100 and see which one is inside.
So in this case, you will have only the cells that are disseminating, not in the other compartment, because you inject them directly. So this we know, we inject adenoma, though we know these cells. They are not the really early disseminated cells, because we cannot pick them.
Thanks.
Julie, there are 2 questions on the chat, and sorry, as I had a problem with the microphone, perhaps I didn't hear if a question has been already asked. Have you looked at the intestinal vascular barrier, which is disrupted in CRC patients and then leads to the translocation of intestinal bacteria to the liver?
No, very good point, but no, we haven't looked.
And the second question, I like it because I wanted to ask you, then does liver remodeling involves fibrotic process, and did you examine also the early dissemination in pathological liver with steatosis?
So second, no. And first was, is there CAF? Is there fibrosis?
Don't say that like that.
Fibrosis, no. I know that if you are interested, there might be CAF around. There is a fibrosis. We have done some staining of fibronectin, staining, I can't remember the name, the color, what? Thyroid red, voila. And yeah, there is definitely a fibrosis that occurs.
This is interesting when you have indeed modulation, modifications of the fibroblast which are surrounding. Not yet CAF, but because you do not have cancer cells, but yes.
We have cells
adenomas,
that,
yes.
yeah, so I don't know if they are CAF or not, so
Yeah,
that's
sure.
interesting, yeah.
Any other questions? Okay, thank you very much, Julie.